the University of Buffalo. She's a great collaborator and also friend, and she's really interested in using multi-scale modeling to answer questions that are biologically relevant across different contexts, from cancer to kidney and others. And so we're going to hear a little bit about her research and also how she's been able to connect with experimental researchers and together guide the questions that they're addressing and how to best apply mathematical modeling in those contexts.
All right, thank you for the invitation, Stacy. And this was a fun one to put together because it really is what we do. So I'm going to tell the story trying to connect some dots between computational modeling and experimental collaboration. And my career started with engineered biomaterials. So when I was a graduate student at Illinois, my lab, I was in Richard Brotz's lab, we were building physics-based models for biodegradable PLGA. Dan Pack's lab was doing drug delivery. I was doing the simulations, and that fostered a passion for understanding heterogeneous, porous biomaterials that were changing and evolving with time, and then that led to the kind of tissue-related problems I investigated as a faculty member. But we still do some work with drug delivery materials as well. So the problems that we choose to work on often involve the balance between tissue formation and tissue degradation. So we do a lot with collagen remodeling in different organ contexts. So things like fibrosis, metastatic remodeling of tumor microenvironments, and bone degradation are key things that we look at, as well as kidney and lung fibrosis. So two types of problems that we typically work on either involve kinetics or transport. So in kinetics, we're thinking about the topic of interest could be a biomolecule, it could be a cell, could be an entire organism. So we have some work with bees where we consider them as a whole organism. And we're thinking about the reactions that they go through, the interactions between communities or individuals in these entities, and how they might change state. So state might be active to or inactive to an activated. It might be switching phenotypes through differentiation. It might be not moving to moving or proliferating or some other kind of function, but we think about these through biochemical networks. So some of the work involves, say, pharmacokinetics and thinking about a drug interacting in these reaction networks. We do stuff related to ECM turnover, and then this is our work with bees, but all of it involves some sort of dynamics and some sort of rates of interaction. And then on the mass transport side, this is actually early work with our PLGA microspheres and cumulative drug release. This is trying to represent cell migration through a remodeling tumor microenvironment and the cross-linking of collagen fibers, and that would influence the ability of cells to migrate preferentially along that stiffer collagen. And then thinking about MMPs and cross-linkers changing that environment. And then in the context of kidney, we're thinking about transport across different membranes, but then how those membranes themselves can be disrupted through cell health and through different things like exposure to long-term hyperglycemia and diabetes. We use a lot of different computational methods, and we've been continuing to evolve and learn more and more techniques. So as a graduate student, I focused almost exclusively on what we call deterministic mathematical models using ordinary and partial differential equations. These were the classic tools from chemical engineering. We did a lot with programming and making user interfaces, particularly for either educational purposes or for collaborators to not have to know the code, but to be able to run a simulation and see some sort of output from that. More recently, we've been moving into R and making scripts and things that are useful for our collaborators to be able to extract something from their data, but then get it into a form that we need to be able to run our models from. We've been moving more and more into the biomechanics. We've been using finite elements for a long time for fluid dynamics, but using tools like FEBio and others, COMSOL. We do quite a lot of dynamic systems, but we also have been dabbling more in data science techniques lately. And then agent-based modeling we really started doing as in my faculty role, but we do this quite a lot with lots of different paradigms. So I won't talk about all of these vignettes, but these are the systems that we currently study. And in the eye context, Caitlin Swindle-Riley at The Ohio State University is in biomedical engineering as well as chemical engineering, and she looks at ocular drug delivery and all kinds of different biomedical therapeutics for vision science. And we have an NIBIB R01 collaborative, and then these are trainees in my lab. And the goal is to take her bilayered capsules, so both microparticles and larger microcapsules. The original data from her lab is using polycaprolactone and chitosan, but we're now exploring some different faster biodegradable materials. The chitosan or the PCL is just not degrading fast enough to use it in the ocular realm, but we think about these particles or capsules and trying to improve designs for long-term delivery to the back of the eye. So we want to think about these implantables, we want to model the drug release from these devices, but then also we've been doing simulations of if we have an injection bolus, what's the distribution in the eye, and what's the concentration that reaches the back of the eye, as well as if we put our implantable device coupling the release rate from that biopolymer or multi-layer polymers and how that then spreads across the eye. And then we've been able to come up with not just using their data to fit parameters for the release from a drug, from the polymer, but also trying to come up with these design spaces. So Chris mentioned rational design earlier. We're trying to, you can't really see the axes here, but this is changing the thickness proportional to the original thickness of Caitlin's experimental work, but making it now thicker or thinner, second layer. And then this axis is making the inner core larger or smaller from the starting value. And so being able to say, okay, there's not a lot of change if we're doing more thicker layers of polycaprolactone, but if we make the chitosan core, that's the bigger determinant in this drug release rate. And this y-axis is, or z-axis is days that the therapeutic is being released over a therapeutic rate. And so we want to maximize that. So we can use these for being able to rationally design with minimal experiments, better bilayer capsule design. We also, in the bone context, this is work with Brenda Smith at Indiana University School of Medicine. She was previously at Oklahoma State, where I was. This is a real example of team science that I love working with Brenda, and I want to continue to work with her forever. And she saw the value of what we were bringing from a multi-scale systems biomedicine perspective and said that she needed more than just the snapshots of samples and different, she does a lot of mouse models for giving nutritional supplements and looking at their impacts on bone health. And so she takes a lot of different tissue samples, but she knows that those tissues are interconnected, they're in living animals, but that there's dynamics involved that she's not capturing. It's expensive to do all of those analyses at lots of time points, but trying to make sense of this, right? Coupling the gut, we're looking at models for osteoporosis, so we have hormone things going on, aging, all kinds of stuff. So one little vignette I want to tell from this is that when we first started, she pointed to this paper on the left that was someone else's experimental paper who had a conceptual model of what they thought the hypothesis, or that their data supported a hypothesis of how a certain nutritional supplement was influencing through the immune system the formation of more bone, right? So we built a mathematical model that could confirm that hypothesis with the data that was in that published report. From there, we were then able to design new experiments with Brenda and have convincing evidence for NIH to be able to get new funding for those next set of experiments, where we now say we're going to look at a different molecule, a different type of nutritional supplement, and we're going to look at this in healthy and estrogen-deficient animals, and we're going to measure lots of things related to the immune system, and we're going to look at these different dietary supplements. And we could actually have this body of work now with different experiments we've worked with, characterizing bone, gut, immune system, transcripts, et cetera. So now we're analyzing her data in lots of ways, and the animation's kind of been, that one didn't get animated, right? We do things related to our classic mathematical hypotheses, where we say, we think that this chemical is interacting in this mechanistic piece, so let's describe that interaction with a mathematical rule. We also have been looking at bioinformatics of RNA sequencing data and coupling with our bioinformatics core on our campus, and we're able to tie some things with gut markers to bone health, which is really exciting. I won't go into all of that detail, but really exciting about that kind of analysis. And we're also now doing some more data-driven, like there's lots of things happening and some of it cannot obey the mechanisms we like presupposed. So trying to do more data-driven to figure out which things from all the many measurements we have are the strongest predictors of different kind of outcomes so that we can then focus in on new experiments about those predictors. And then the final story I'll briefly tell is related to our cancer work. So this is work that I was interested. I wanted to think about the remodeling tumor microenvironment. So my lab built these simulations related to how cells would migrate out from an organoid or a primary tumor while there was remodeling of the material around. And we explored things like what's the starting collagen porosity and stiffness nearby, and we could explore if it was randomly oriented fibers that had a lot of like uniform or heterogeneous, or they were uniform at the same concentration, and how that affected migration patterns, and start to think about spatial statistics and things related to these patterns. But I showed this work at a conference. I've shown this at a Termis meeting, I've shown this at a tumor microenvironment meeting, lots of places where experimental people who were doing the kind of things I was interested in were at. And that led to meeting Mike Dwinell, who's at the Medical College of Wisconsin, and he's a cancer biologist, immunologist. And so now we have an active partnership where we can combine the immune things I'm interested in with the kind of experimental techniques, both animal and clinical work that he does. So now we've been moving more into spatial transcriptomics and being able to analyze that data, being able to know how to use that kind of spatial data, some more mathematical tools like spatial and topological data analysis. So it's not just how many cells there are, but metrics for understanding the spatial distributions and being able to characterize those. And so those are new techniques from my lab, but we're really trying to stay up to date with the best knowledge there in the spatial biology, as well as putting that information into our agent-based models to have the evolution of the starting conditions of where the cells and their maybe what transcripts they experience in the background for, let's say, ligands, how they're then responding to ligands and moving through the environments and trying to understand some biological with Mike. We're also building for in vitro experiments, so where we have a lot of data from Mike's lab and from others, we're trying to build high fidelity simulations of that environment so that we can put in properties for those cell migrations so that we can put the cells back into realistic biomaterials and let them simulate, but we build the parameters based on the transwell data that's more readily available. So those are kinds of things that we've been tackling and the kinds of methods that we use, but very interested in spatial things, very interested in dynamic things, and I call it multi-scale modeling too, but I'm a little bit larger link scale than what Ravi does. I'm thinking more like chronic diseases and tissue level, not molecular level. So I'm not gonna look at what molecule binds to which thing, we're looking a little bit larger than that, but they're all examples of multi-scale modeling. Okay, so that's the last that I have. I'm gonna stop there. Stacy asked me to be brief, so thank you for your attention.
One or two questions, if anyone has any. Yes, please. Do you want to go to the mic?
So whenever you're doing like the simulations and stuff, so like, do you end up, I guess you did talk about COMSOL a little bit as well. So I guess my question is like, do you think there's like a sweet spot in the middle where you're doing like Abacus, COMSOL, all of those stuff, and also on one end, then you're doing like...
So we like to not reinvent the wheel.
Okay.
So if somebody has already made an amazing tool, we are not starting from scratch and wasting years to revalidate that tool. So COMSOL is an example of a software that does fluid dynamics very well and does biomechanics and structural things very well. But there's agent-based modeling tools and molecular modeling tools where people have put a lot of effort into making those algorithms work. But sometimes the algorithms are not there for what we're trying to do, or we might be bridging different scales and we might be trying to couple. So we've got some stuff right now where we're trying to couple some of our reaction-based models in MATLAB or Python with something in FEBio that is kind of like COMSOL but open source. So we're coming up with ways to couple those and run them outside of that. So in those cases, we're building new algorithms. But we try to focus more on the application rather than new algorithms. But we're not opposed to learning new algorithms or creating them as needed. But it's a different kind of outcome from doing new algorithms. So.
I guess the other one also is like, especially like, I guess at this point, like if you want to run something on like an HPC environment, so I guess you might be limited with the platform that you're using, right? So.
It depends. So we do a lot of work that's not, one run might not be super long and need HPC, but we might want to do, let's say 15 replicates of the same setting, but then we might want to sweep through different parameters. So we consider it embarrassingly parallel. Each different run, we just send to a different node on the supercomputer. But each one may only take 1 or 2 hours. It's not like it's going to take weeks to run one of our simulations, but we might use the power of high performance computing to just run high throughput computing, much like you might do automated high throughput experiments, just so that you're not having to do them serially. That way it's a cost or a time savings for us. So that's the way we tend to think about them. And so we can run some of those on GPUs or on the HPC, but not have to parallelize some of those programs that are inherently not parallel themselves.
Okay, thank you.
Yeah.